Hey guys, this is part two, the comments that I'm adding to the chapter 8 notes. And on the last episode there, so to speak, I was talking all about the light reaction and how Photosystems 2 feed into Photosystems 1. Eventually, you're going to create some ATP here in the electron transport chain and end up with a couple of NADPHs. You're splitting water originally to end up with some hydrogen, which then is bonded to the NADP plus, and that's how you get the batteries charged up there. So if you missed this one, go back to slide 1 or the video 1 and take a look at it again. The next slides on 17, 18, and 19, they just go over what is on the slide itself. And so I'm going to go jump to slide 20. And slide 20 here is just talking about the light independent reaction again, where it's saying that oxygen is being released but as a byproduct after water is being split. You're transporting those hydrogen ions to the stroma, to the thylakoid membrane. Okay, and that's the thylakoid membrane is going to be then releasing these to the NADP plus. Okay, so then as hydrogen ions inside the cell react with ATP synthase to create ATP also. All right, so you got hydrogen ions that are being used to create the NADP plus. You got hydrogen ions and electrons that are also being used to create the ATP. All right. So that was on slide 20. On slide 21, all I want you to do is to add this video at the very bottom here, and to get this video link again from going to the assignment in week 1, semester 2, week 1, go to the Nearpod photosynthesis and notes assignment, and this link will be right in there. Remember to, when you copy and paste it in, to hit enter, and that will make it a live link. If you don't, it's not going to make it a live link, and you can sit there and click on it all day. If it's not blue and underlined like this, you go to just to the right hand side of the 4 and then hit enter, and it will make it a live link. Okay, the next slide, slide 22. This one here is talking about where are these things actually taking place? Where's the light reaction taking place? Where's the dark reaction taking place? And I added these boxes here, the thylakoid membrane and the stroma, as to in these boxes here, so that you can tell light reaction takes place in the thylakoid membrane. Dark reaction takes place all around all those membranes in the stroma. Okay. So moving on from that, slide 23, there's no additional comments to slide 23, but we get to slide 24, and I've added this picture here at the bottom. And again, this picture is found on the in that assignment, that Nearpod photosynthesis and notes assignment, found in week 1 of the 2nd semester. Just click that, add it to it. You may want to add some notes and comments on this one. When I went over this picture, and I don't know why it wigged that out, I was on the right tool. Okay. Yeah, let's go right about there. That's good. What I want you to get out of this picture is not so much all of the names, because they are kind of complicated, but just the fact that you're going to take some carbon dioxide and you're going to take another molecule. You're going to add these things together using an enzyme called rubisco, and it's going to change the shape of this thing. And for every single step, it changes the shape. It tears it apart and reassembles it, tears it apart and reassembles it. Every time it tears it apart and reassembles it and just adjusts it a little bit, it's using energy. And that's where that energy that you made in the light reaction is being used. It's used to manipulate these molecules, to tweak them, to move them, to reorganize them. going from like a 5-carbon molecule down to a 3-carbon molecule, from a 3-carbon molecule in one configuration to another. You think of this one maybe as a triangle, this one here is a circle, and this one over here is maybe a polygon. Okay. Excuse me. So you use an ATP to do this. And when you do that, it's going to lose that phosphate. Well, the phosphate's got to go somewhere. And so usually it hooks up to the end of these things because it needs another bonding site. And as it goes through and keeps manipulating, sometimes it needs one, and sometimes that new configuration needs two. And so once it gets two, it's like, all right, what do I do with both of these? Well, then I'll knock it off again and come down to one. So eventually what you need is one of these 3-carbon with a phosphate at the end. So you're trying to get this thing down to what's called a glyceraldehyde, aldehyde 3 phosphate. In other words, there's 3 of these. The configuration is a glycerol and an aldehyde, which is in a certain configuration. If you've taken some really up there chemistry and organic chemistry, you would understand all these things. And again, I don't need you to. I just need you to understand that they're changing shapes. And as they're changing shapes, they're getting closer and closer and closer to the shape of what sugar really should be. And one of these 6, this 6 means that there's actually 6 of these molecules. One of these 6 molecules is going to get spun out of there. And the rest of them are going to be used as starters for the next batch. So think of this as that bread that you use where you need a starter piece, so almost like a sourdough bread or a shepherd's bread, where you got to have a little bit of that bread to get the next batch going. You got to have a little bit of these molecules to get the next batch going. All right. You don't just put in a bunch of carbon dioxide in there, break it all apart, and boom, you reassemble it into a sugar. It's a slow process. And you're going to need 2 turns of these. So you're going to have to do this thing twice to get 2 of these molecules. They call them G3Ps because it's 3 carbon molecules that are the glycerol aldehydes with a phosphate on there. You're going to need 2 of those. They then combine together, and as they combine together, they lose the phosphates. These phosphates are then used up here in the ribulose biphosphate, and they're also used as they go back to the thylakoid to be used for the NADP plus, and actually they're used for the ADP going to ATP. Sorry, phosphates, ATP. NADP plus uses hydrogens and some other electrons. Okay, so these phosphates then go back to the thylakoids, go back to the light reaction, used to recharge the ATP battery, and then to be used all over again. So once you get 2 of these molecules, then it's reassembled, retwisted around, and manipulated into a glucose molecule. A true glucose molecule, they call it a chair configuration. It's if you were doing geometric shapes, it would look like a chaise lounge chair, where the back of the chair would be a triangle, and then the seat of the chair would be a square, and then the leg part that supports the legs would be another triangle coming to a point. So you got a point going to a square seat, going to a point. So it goes down and across and down is what an actual glucose molecule looks like. But you guys don't need to know that either until you get to organic chemistry, which then all of this becomes a little more fun. For right now, you don't need to know all those steps. What I'm trying to get you guys to realize is the energy that is produced from the light reaction is transferred to the Calvin cycle here and is used to manipulate and rebuild these molecules until it finally gets down to a form like glucose. Okay? And it takes a lot of energy to do that. All right, so as we're moving along here, slide 25, 26, 27, 28, and 29 are all just slides that have really good information on there. You should know what's going on in there, but I didn't add anything else to it. And the last slide here, slide 30, this goes over some factors that can affect photosynthesis itself. It's got to have a good water supply. If it doesn't have a good water supply, it's not going to produce. The temperature's got to be the right temperature. It's got to be somewhere between 0 and 35 degrees Celsius. Anything above that or below that, it's not going to produce photosynthesis. And it's got to have the correct intensity of light. In other words, it's got to have blue light, red light, orange light. It cannot have like a ton of green light and none of the others because it's not going to work. All right. The comment I put down here at the bottom is just a transference of or transformation of Celsius to Fahrenheit. So it's a correlation. Most of you guys don't understand how cold or how warm 0 degrees is in Celsius. Most of you guys think, oh my God, that's 0 degrees. That's like cold. Well, it is cold. It's 32 degrees. It's freezing for water. 35 degrees Celsius, though, is 95 degrees Fahrenheit. So it goes up quite a bit. So I think 35 is definitely on the toasty side. 0 is a little on the chilly side as far as trying to photosynthesize. If you're at freezing, that means water is hardly doing anything. It's not going to move around too much. Once it starts to turn to ice, well, the plant's in big trouble. So 32 degrees is questionable. At 35 degrees, it's also questionable because it may be losing a lot of water at that point. So somewhere in between there would be a better number. Somewhere probably in the mid to upper 20s would be a better number. Okay, so I hope that helps. Make sure those are on your slides and you've checked them over and you studied hard for the upcoming test. All right, thank you.